Good morning, microbiology students. I wanted to make a quick video explaining some of the things in our notes since I'm not going to be there today. Our goal is to really start looking at um, cells and microbes, and I think we have to start at the foundation with our understanding of this. We just finished up history of microbiology. We want to spend a little time looking at, really the four macromolecules of life. And so let's get started here. Change this to red quick. So we're in chapter 2 of your book. You'll see your assignment sort of coincides with what we're doing here in the notes. In microbiology, we study microbes, but what are microbes made of? I think that's where we have to start. And if you remember from your biology classes, you remember that, this is a prokaryotic cell. It's got some DNA in there. It's got these little things in here, these little guys, all these little structures, right? And so you probably threw around terms like organelles, right? Maybe nucleus, maybe cell membrane, ribosomes. And you're right; those are what microbes are made of, right? These little individual cells. This is a bacteria cell we're looking at, but I think we can go a little deeper with this and get beyond, some of those organelles. What are those organelles made of? Well, let's talk about that. Let's start there. So, um, as we look at what living things are made of, we can really boil it down to these 6 elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, or you hear me refer to it as chomps, right? There's a lot of different elements on that periodic table, but these are the 6 most important ones for life. And in fact, 96% of a living thing is made of just these 6 elements, right? So these are the 6 major ones that we need, okay? Now, these 6 elements are going to combine to make these things called macromolecules. And a macromolecule is just a big molecule, right? And made of those 6 elements. And so the ones that we look at, or I guess the 4 that are out there, are carbohydrates, right? And you've heard of carbs before. You probably have eaten carbs before. Lipids or fats. Lipid is a fancy word for fats, okay? Also something that we find in our diet. And then protein, right? Which is also something you find in our diet. And then nucleic acids. And so when you hear about these 4 macromolecules, you typically think of them with food. And that is true. We do eat these things. They are a part of our diet. And I mean, if you really take a second just to step back and think about it, it kind of makes sense that the stuff that you need to eat are the same things that cells are made of, right? Our goal is to make more human, right? If you're, growing, and you guys are all young adults, so you're growing, you need to take in the things that your body is going to need to make more cells. And those things are carbs, fats, proteins, and nucleic acids, okay? And so when we talk about them, we really look at things from a bacterial standpoint. And when we say macromolecules, they're either food or structure. It boils down to those 2 things. So if we're talking carbohydrates, it's either food or it's structure to make new cells, right? Whether it's a structure within the cell, whatever it is, okay? And we also talk about, and I actually heard, I was walking by Mrs. Richards' class. She was teaching organic chemistry, and she was talking about carbon with her organic kids. And she was talking about the fact that, most things in organic chemistry are carbon based, and that's because carbon's like the ultimate Lego, right? This thing is the ultimate Lego. It can bond up to 4 times, right? It wants 4 more electrons, so it wants to bond 4 times. It is the ultimate Lego, and it'll build all kinds of amazing things. And we'll continue to see that, right, as you talk a little bit of biochemistry, okay? So where are we at right now? We know that we study microbes, and we know that microbes can be really boiled down to be made of these things, right? Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And those things are made of, or make up, I should say, make up carbs, lipids, proteins, proteins, and nucleic acids, okay? So where do we actually see those things in our cell? Well, we'll see this inner layer, number 1 here, is called the cell membrane. That's actually made of lipids. We'll see that here in just a little bit. This genetic material in the middle here, that squiggly spaghetti looking stuff, that is the most famous nucleic acid, deoxyribonucleic acid, okay? We see all these other little things here. We're looking at actually this yellow layer here is made of a carbohydrate. It is the cell wall, okay? And bacteria have cell walls, plant cells have cell walls. And there's a bunch of other little things inside of here as well that are made of these proteins. And so we'll see these things throughout the cell come up. And I think if you understand sort of the structure and properties of these different macromolecules, everything's just going to make a little more sense as we move forward in this class, okay? Before I get too carried away, I want to make sure I talk about building and breaking polymers here, okay? We consider these macromolecules polymers, okay? A carbohydrate, a fat, a protein, those are polymers. They're made of these big, huge, complex molecules, okay? Well, these polymers, you've probably heard this word in like plastics or chemistry or whatever, are made of these things called monomers, okay? And a monomer is simply one building block, and a polymer is a chain of those building blocks, okay? You'll see me in class. I tend to use, I don't know, you call them like these connect beads or whatever you call them when you were a kid. We've all played with those when we were kids. Think of one of those as a monomer and a chain of those as a polymer, okay? And a chain of those blocks is going to make something that our cells need, okay? And so it's important that we understand, how do you make these polymers, and then how do you break them down and then use them to make new stuff, right? Because cells are always making new things too. And so you may not like the chain like this. You may want to make a different chain. Well, you got to know how to break those things down. So that's what we want to talk about here for just a second. So how do you build polymer chains? Everything revolves around water. We know water's important. We just don't really quite understand what our cells do with water. And so we want to shed a little bit of light on that. So when we build a polymer, we actually remove water. And so think about if you were dehydrating something, right? Imagine if you had water and salt together. Removing that water is going to help that salt recrystallize. It makes that salt bigger. And that's kind of the same way it works here. But let's talk about where that water came from or where it's going. And so let's say we're talking about building a polymer chain, and we can see that down here. You got all these monomers put together, and then we typically cap any sort of polymer with an OH on one end and an H on the other end, okay? But let's say I wanted to add this monomer to this chain over here. What do I have to do? Well, the first thing is, if you want to do that, you're going to have to take this hydrogen cap off of the polymer chain, and you have to take this OH cap or hydroxide cap off of the monomer. And if you can do that, you can take off that H and that OH. Well, one hydrogen, another oxygen, two hydrogens and an oxygen, that makes water. And so you literally are removing a water molecule, essentially, half from the polymer chain, half from the monomer chain. And that will allow then those two to bond and link together, and you'll form this bond here and make that chain that much bigger. And so that's how you build a polymer. You have to remove water to be able to add to it and free those bonds up, okay? And the opposite of true is breaking a polymer down. So if you want to break a polymer, well, you do the opposite. You add water, okay? So instead of removing our water now, we're going to add water. Water is a polar molecule. It has a positive end and a negative end, okay? It's a polar molecule. And so because of that, water tends to just kind of tug on everything. And eventually, if you give water enough time, it can keep tugging on these bonds and eventually break them down, and that water will dissociate. You get a hydrogen capping one side and an OH capping the other side, and so you can break that polymer. And that's why, water is really important for digestion and living things. It's great. It's a good facilitator for helping break things down inside the body. Let's take a second and talk about carbohydrates, our first macromolecule. They're made of these simple sugars, basically things that have carbon, hydrogen, and oxygen in this ratio of 1 to 2 to 1. And a simple sugar is simply usually a little hexagon or a pentagon. Here's a simple sugar down here called glucose. Okay, you've heard of glucose with plants before. Really, their job, our carbohydrates in our cells, their job is for fuel, right? They are what we use for energy, but it's also for bacterial structural support. When we start talking about bacteria, we'll see that carbohydrates are actually important for bacteria cells to build their cell wall out of. It's made out of this stuff called peptidoglycan. We'll talk about it down here. And so I want you thinking, when you think carbohydrates, I want you thinking fuel and structure. Okay? And when we talk about carbohydrates, we typically talk about them as mono, di, or polysaccharides, either one sugar hexagon, two sugar hexagons, or many put together. And so here you can see a mono. Over here you can see a di. Oops. You can see that disaccharide there. In sucrose, that's the stuff you put on your cereal. And then here you see a polysaccharide. Boy, goodness sakes. A polysaccharide there. It's just a big long chain of those hexagons, okay? And sort of the rule is this. Any living thing, when you are needing fuel, no different than when you're building a fire, you can't just take a big, huge stump and start that thing on fire. It's not going to work very well. What you're going to need to do is chop that thing up into much smaller pieces, probably add some kindling and some really tiny bits of wood if you want to make a fire. And the same is true in our bodies. If you want fuel, you need to get whatever you ate in its simplest form, right? It's gonna be usable in its simplest form. And so our body prefers monosaccharides, okay? And these tend to taste sweet, glucose, fructose. Your body can use those, and any cell can use that as more of an immediate fuel. You need energy now. Those are the ones you want to eat. Things like disaccharides, sucrose, lactose, maltose, we tend to eat these. They come combined like that as a pair, two sugar rings put together. It doesn't take much. You break those two sugar rings apart, and then you're in its simplest form. That's a pretty quick source of energy as well. But these polysaccharides, they take some time to break down. And so polysaccharides, those big long chains you can see, we need a lot of time to break those down. And so like pasta, things like that, we tend to have to eat those. Anything with starch in it, you tend to eat those hours in advance, sometimes the night before. You're going to need that energy. So if you play sports, you probably know pasta is a good thing to eat the night before a race or an event, but not an hour before necessarily. You don't have enough time for that to get broken down typically. So and our body has enzymes to break things down oftentimes, but I think I want to highlight a couple things here. This peptidoglycan layer is going to become a big deal when we talk about cell walls, so make sure that you key in on that a little bit. In cellulose plant cell walls, we're going to read an article on making ethanol out of some different types of plant materials. There's a lot of energy stored in those hexagons. And so if you can figure out a way to break those bonds and get that energy out and release it, you have potential to, have a fuel source essentially. And so you're going to read something about that probably on Thursday, looking at different types of carbohydrates. But, carbohydrates, they work great. You look at things like a tree. It's made of lignin, right, and cellulose, and those are really hard materials. Carbohydrates can be a great structural material, in living things. Trees don't have bones, and so how do they stay rigid? It's really these polysaccharide chains linking together, making these really rigid structures. Okay, so carbs, fuel, and structure. Lipids, our next one. They're building blocks, or their monomers are glycerols and fatty acids. When we talk about lipids, it's usually as fats in food or in phospholipids. And that's really where I want you guys thinking is phospholipids are the things that make up cell membranes. Here's a phospholipid down here. It's got this head and tail structure. You can see it. And that's what makes up all of our cell membranes. Bacteria, humans, doesn't really matter. It's those head and tail structures. And, you can see here that they're hydrophobic. And to be hydrophobic means that you're afraid of water. And so those tails will sit and act like a barrier on the inside there. You can see all those tails orientated towards the middle. Oil and water don't mix. And so those tails really repel water well. And so, when cells are trying to keep water from dissolving them, water dissolves just about anything. And so how do you keep water from dissolving our cells? Even when your cells are bathed in water and you're 70% water, it's really those phospholipids, that macromolecule there that really helps protect your cells and keep them from getting dissolved. And it really helps regulate what goes in and out. But the interesting thing is soap can break those lipids down. Soap's great at breaking down grease and oil and things like that in our dishes. It can also work to break down cell membranes. And so that's part of the reason why we use soap with bacteria is that soap can go through and damage the cell membranes of those cells and, kill them in the process, which is good because, helps keep us healthy. So one of the questions I sometimes get is, well, if I have cell membranes, why doesn't soap destroy my cell membranes? And we'll talk about that. You have this top layer of skin cells that behaves a little bit differently. And so we'll look at that in class together. Okay. But make sure you understand phospholipids are what make up cell membranes. And in addition to that, there's all these other proteins like channel proteins that let stuff in and out. You have these receptor proteins. Those things are going to help cells signal with each other. That's how cells communicate to each other. What those receptor proteins they have, it tells you if cells can recognize each other or not. So, proteins, carbohydrates, lipids, they're just huge in terms of cell structure and helping cells function the way they do. And in our understanding of that, the more we understand about these macromolecules and where they're being used, that's also how medicines are able to target certain things. They can find, what are some of the structures within cells and use those to help design medications to destroy bad cells or things like that and help get us better. So lipids. Nucleic acids, they have 3 parts. They're made of these things called nucleotides. And the 3 parts of a nucleotide is a sugar, a nitrogen base, and a phosphate group. We had talked about CHOMPs, CHONPS. We can see all of that in play here. That's what DNA is made of, these nucleic acids. Hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Just no sulfur here in DNA. But you can see those nucleotides there in their structure. It's got that phosphate group, that sugar ring, and then that nitrogen base. And when you see DNA, it's usually as like a code A, G, C, A, T. These are each of those is a nucleotide, okay? And so when we talk about really 3 types of nucleic acids, DNA is probably the most famous one, deoxyribonucleic acid. We already talked about those nucleotides there. There's also RNA. It's a single-stranded sort of helix. It's not quite helical. It's more of a single strand. You can see DNA here, RNA there. And then ATP. ATP is cell energy. This is essentially like having cash, right? When you eat food, think of that as like a check, right? If your grandma gives you a check for $100, that check is useful, right? It's great. Thanks, grandma. But it's not useful as is, right? We have to cash that check. ATP is cash. Your cells can use that the check is like the food you eat. It has to be converted into cash first. And you get all that energy out of ATP when you break off this last phosphate here. Okay. And so nucleic acids, we need them for storing genetic material, providing instructions to make proteins. And it really is also used as an energy as our cell. And so, I mean, you're starting to see where these macromolecules are used. And I'm going to say that probably one of the more important ones, at least for now, is proteins. Proteins are made of these things called amino acids, and there's 20 essential amino acids. And so an amino acid would be like one ball on this chain, one bead on the string. It's essentially the monomers, right? And then you get a chain of those. That's your polymer. That's your protein. Okay. And so the function, gosh, you name it, right? And that's why I put down here in bold, for anything to happen inside a cell, it needs proteins to make it happen. So whatever it is, anything that's ever happened in a cell, it was a protein that made it happen. Okay. They're just crucial for helping cells do the stuff cells have to do, whether it's communicating, providing structure, speeding up reactions, regulating what goes in and out. They help break things down or make new compounds. I mean, the proteins in your cells are really the ones doing the work. So they all have a pretty similar structure. They have an amino group, a carboxylic acid group, and they've always got this side chain, and that's what makes amino acids different from each other. I think that's a question I asked you about on your assignment. But essentially, you put a bunch of amino acids together, you get this thing called a protein, or it's a polypeptide. It's going to become a protein as soon as it folds, but essentially a protein. But worth noting as well, all of these are a little bit different, right? We got reds and greens and blues and oranges. Well, the sequence of those is going to make a different protein. And so these amino acids, depending on if you go red, blue, purple, orange, or orange, orange, green, green, yellow, blue, the sequence and order of these matters, because this protein chain is going to fold, and how it folds determines what it does. And so that sequence of amino acids matters. And so if you put these greens on the front end of this thing, it's going to change what this thing does. If you move this orange to the middle, it's going to change what it does. And so keep that in mind as well. I think sometimes we get that confused. So we've really spent time talking about those macromolecules, these things that make up microorganisms and the cells of microorganisms, whether it's the DNA inside of there, the cell membrane of these cells, whether it's the capsule layer, or sorry, the cell wall layer of these that provides structure, or it's the food for these. I mean, we're going to see these things over and over again. And that's one of the things that we key in on in classes. How are microorganisms different from each other? And one of the answers is it's in their macromolecules. Okay. And so we talked about proteins. We know that for anything to happen, you need proteins for that to work. Okay. This just talks about protein structure and folding. I think one of the things that's important to know is that, this protein here, it's basically ready to go, but it's not a functioning protein yet. What has to happen is this protein needs to get folded up. It's essentially like getting the key sort of cut in the special pattern so that you can actually use it to unlock your door. A blank key isn't helpful. Your key needs to be cut in a particular pattern. You got to go take it to the hardware store. They'll cut the key for you, and then you can go use it to open doors. The equivalent of that is folding this protein chain, or this amino acid chain. Once it folds up into this bigger structure here, it gets its shape, and that determines what it does. And so that's a functional protein there, all that folded up sort of squiggly mess that you're seeing there. This is also a folded protein. That is a functional protein. Well, can you make a protein non-functioning? Yeah. Heat and certain chemicals will cause that protein fold to unravel itself. We call that denaturation, or denaturing a protein. When you unfold that protein, you basically make it useless again. It can't do its job anymore. That key is no longer useful. It can't open any doors. And so when you apply heat or chemicals inside of a cell. This is also why, heating your body up gets kind of dangerous. If you unfold your proteins in your body, it can cause serious damage. And so, we always take fevers and things really seriously. We're really worried about your proteins denaturing is one of the big things. And so I just wanted to talk about that briefly. Again, proteins, they're going to allow our cells to function. I hope you get an appreciation looking at this diagram. All the little proteins that make all the little things happen inside of our cells, they're just crucial inside of the cell membrane that you're seeing here. Again, another diagram there. You can see two cells kind of coming in contact and getting ready to communicate. Cells, this is how they talk to each other. This is how they recognize each other. This is how your immune system recognizes who to kill, who not to kill inside the body. It's all in your membranes, okay? I want to save our conversation for enzymes until Thursday. And so you can be working on your assignment. I'm going to talk about enzymes on Thursday and we'll go from there. But I think that's enough. I want to take and really devote some time to enzymes. And so that's where we'll stop today, if we can.